Hello, good morning, good evening. Thanks for listening. This is a Medical Cannabis Show, and I respect the law, and I hope you do too. Today is an open topic, and we'll see if we can get to some sort of topic. And that's generally waiting on the chat to come in, and some questions will come through, and then it happens. So if you were watching this back, you can listen to me try and make some sense out of what's going on, which I don't know, or you can fast forward to some slides, and that will be when there'll be a topic and we can start our discussion. And if it only goes for 10 minutes, that will be when you'll know that there won't be any real topic today. And that happens every now and then, which I don't mind, because some of the topics I just get excited on and can talk for a long time. And I look up and think, holy moly, look at it. There's already been like an hour and a half going or something. And I don't mind doing an hour, but sometimes they go for 2 hours and even 2 hours and 10 minutes and things like that. I think that was the longest one. So I'll just wait until 2 minutes for things to start. And then I'll start my little ramble again. So that's 30 more seconds. And we'll see what happens. So another 20 more seconds to go. And then 10 seconds. This is interesting. What comes to mind is the weather. As you know, that's one of the topics in the world that's been most talked about if you look it up. So I can only think about the different hemispheres that we're going through at the moment and the grow conditions, what the soil's going through, and all the microbes are loving it. In the northern hemisphere where it's warm, and the southern hemisphere going, come on, it's too blooming cold. So hopefully there'll be some chat soon, and then I can start talking about something that probably interests you more than just stuff that's going on with me. And all I can think about really is in the middle of summer in the northern hemisphere, in the middle of winter in the southern hemisphere, bit of a tongue tire, and what's going on in those conditions. This fellow I understand. Hello, Main Green. Nice to see you, mate. He'll be getting ready to transition into the freeze soon. I'm loving the heat at the moment. And in the southern hemisphere, yeah, they even said there might even be some snow in the mountains in Australia. Can you believe that? Yeah, well, it doesn't snow in Australia. Well, there might be some, they reckon. Well, there's ski fields that, there's ski fields, but they said in the other mountains there might even be some. The weather's going on about with the Bureau, so it's pretty cool if you're into that. And Main Green says, yeah, heat and rain till October, November. Yeah, heat and the rain, getting out in the rock fishing, doing the cool stuff. That's the way. Hello, Shipwreck. Nice to see you, mate. Just discussing the Northern Hemisphere and how the heat is going and how you Northern Hemisphere folk would be enjoying it. A lot better before don't even speak about that. Those seasons coming up soon. But yeah, in yours, you should be fully into veg. So things should be pumping along. You should be having no deficiencies. Things will be getting ready to turn very shortly in the next few weeks. No, that's in September it happens. So things could flip from about mid-August. Depends on your position, really. If you've got trees shattering it, you might actually start flipping now. Or if you've got like train wreck, which turns a little bit early, or some autos, that could also be happening early, but in general. Shipwreck says, hope everyone's doing well. Nice one. Thanks for your nice words, mate. Hope everybody is also. I haven't really got much of a topic yet. If you've got something, put it in chat, please. If you've heard anything that's weird through the week, or you want some clarification on something, there might be a myth that you think, wow, is that really true? Does it really blimmin' do that? Or something or other. Controlling the plant. You can control it from various different ways. Main greens eating mushies, having medicinal fun. That's the way, mate. Very good. Nothing wrong with them. Do you know you can actually get them prescribed in Australia now? Medicinal mushrooms, medicinal psilocybin. It's, what does it cost? It's like 8 to 25 grand rings a bell. So you go to these clinics for a week and they sit there, and then they got, because folk that's done it previously, that you have to sort of get a bit prepared, because in the midst of things you're a bit sort of loopy, or things could happen. So the government's taken it on, and they've gone and put all sorts of nurses and physicians and everything around. It's like you see in the ayahuasca, the Amazon Valley, how they've got those camps. It's kind of similar to what the Australian government's done, but because everything costs top dollar, they've gone and everyone's going to make their bid. So that's why it's not cheap, but it's still available. So yeah, because it works so well. Medicinal mushrooms, holy mackerel. Well, cubensis is the only ones I support, because those ones are you can regulate it by weight, the dosage by weight. There was actually a mycologist that quizzed that on LinkedIn a while ago. He said to me, no, you can't. And I said, well, I'm just speaking from experience, because I've done it, I don't know, 100 times at least by that. And actually, Bane Green, do you agree with that? The psilocybin, the cubensis genus can be regulated by weight. It's not just how strong its psychoactive ingredient is inside of it. Do you find that too? Because that's why I only support that one. Main Green says Big Pharma trying to take over in the States. Yep. Everywhere. They just want to monopolize everything, unfortunately. Even in Australia, they've banned the vapes now. So you can't use your vape. You can use your medical one only, but it's going to probably be hard to get through, and they'll just, all sorts of this and that. So all of a sudden you just can't get them anymore. You're not allowed. So it's, just putrid. Hey, GMF. There you go, mate. I see ya. Yeah, roughly 4 to 6 grams. Holy mackerel. I used to have 0.6 to 0.7, and that used to be my event. That's what I used to remember. And it wasn't a microdose either. That was a full-on, how you going, rad fun. Stereo loud, and the TV was on visual effects. I downloaded all these visual effects. Actually, was it downloaded? Yeah, I was downloading back then. It was back in those days. I still had VHS tape system stuff too. But yeah, the medicinal mushrooms seems to be on the topic today. No real questions so far, but it's something that's really works. I don't speak about it enough. For those who, it's legal in Australia to actually have the spores. You can, as long as you don't germinate them, similar to probably what the round-looking one that you're not allowed to cut. So you can have those and grow them, but if you cut them, you're in trouble. Same sort of things. You get, oh yeah, we were just talking about the amounts. I like about 0.6, 0.7 I used to have. I used to be pretty cool. And Main Green was saying he gets colours and stuff off about 1 gram, but he likes shadows and stuff, and the more hardcore things that twist and they morph, and it's not. I don't really like when it gets into that stage because it's sort of too confusing for me and seizure-like. That's what he likes. I understand. Fungal soil is good for flower in medical cannabis. Bloody oath it is. Fungal, yes, you can have a fungal dominant even, and that's, you'll know if it's fungal dominant because your pH will probably be below, because 5, because not in all cases, because in forests, Pacific Northwest, it's fungal dominant. Just have to look under the surface, and that pH is 6.5 odd. But generally, bacteria, they don't survive in below 5. They can, there's acidophiles and stuff like that, but generally, that's what I was meaning by getting that. And yeah, it's handy actually. It's nearly an asset to have. You need to have your beneficial fungus as well, because it's an extension of the roots. So they intertwine, as you know. The mycelium generates a lot faster than the roots of the hairs. So as it extends out and starts to grab and break down nutrients, a lot of them solubilize phosphorus. Sorry, that was pretty cool. So by releasing that phosphatase enzyme, they can break down phosphorus and make it available to the plant. So then it goes into the roots, and it's the extension of the roots by absorbing it through the fungus, and it's releasing back to the roots because it can travel along the fungus. One of them, actually, while you're onto the good fungicides, fungus? I use metahisium and asoplay, and that also is a biological insecticide. It kills a lot of insects as well, larvicides, you can get stuff like that too. GMF says, all right, what do we need to mushroom farm? Okay. Hey, look, there's 669L Monte Carlo. Here you go, mate. I'll see you. If you come in a few times and talk and stuff like that and you seem all right, I'll give you that little monitorization thing. GMF says, what do we need to mushroom farm? Main Green will be able to help good here. We need the right substrates to grow the fungi in, and the right temperature with humidity. So once you've got the 60 to 80% humidity, which is pretty high, and the temperature is around like room temperature, mostly, it varies a lot, but that's what you want it to be to culture. And it should start and multiply. So if you've got to give it the right feed, so as long as there's macro and micronutrients in there too, it should be right. So by giving that on a good base of like a cake, like what was it? What I did a few weeks ago? It was on seaweed kelp meal. So the extracts of the kelp meal, that's just perfect. So all I did was I was making a liquid kelp mix. So I was flushing it and soaking it, and then after I squeezed it out and had the cake, that's the after results of it, the material, that sat there in my strainer for a while, for a few days, and then it started to fur up. And I thought, what a shame, I didn't inoculate it with the one that I wanted. It was inoculated from the air, and I don't know what that was, what species. You can probably know the genus, because colors is a good thing with your beneficial fungus. But I won't get sidetracked. All right, so what do we need to mushroom? So just a good base like that. What else I used to use? Like seed, wheat straw, even grass clippings, but grass clippings isn't that sterile, so you might have to pasteurize it first, because if you don't pasteurize it, you're going to get those spores that are already in there. They're going to germinate, so you don't know if you're growing the ones that you want. So by pasteurizing it, it'll sort of sterilize everything, and then you can inoculate it with what you want, and then you know that that's going to germinate and that's going to multiply heaps. So within a few weeks, you'll pull it apart, and you'll see the cake in the middle of the spores that might fluff out even, and that's what'll start it up. I'll go down. There's some more comments. I'll see if that helps, or if you want me to elaborate more. Been wanting to start one for a while. Yeah, a good one, a good option too, if you haven't got those types of ingredients, is you can go to your local hardwares, and they've got mushroom kits, and a lot of them, they're not inoculated, so you can get your spores that you want and use those mushroom kit substrates and just learn from that. So you might only pay $20 for a start, and then you've got the square box, and you've got your substrate already pre-established for you, and then you just got to inoculate it and put it in the right temperature, and just keep it a bit wet, and that's it. And within no time, you'll be having the white mycelium, or it might be white, depends what species, what genus you have, but in general they're white. But then you'll have the fruit. The bodies of the mushroom will be coming off that afterwards. Once you get the colder temperatures, generally it's 4 to 14 degrees Celsius, which is around 38 to 14. What's that? 55 Fahrenheit is you want for the bodies, for the mushroom bodies to start to fruit, and that's why you'll see in springtime, and well, it depends where you live in the world, but yeah, that's when they're going to start and fruit out. So they're sitting underneath, just waiting for those right temperature conditions, and then they throw the little tops out. Ground mushrooms is quite easy. Yeah, speaking from a person that's done it for the last, how many, 15 years, and done it really, really well with heaps of different ways. Easy for you to say. But it is in general, I agree. Once you got the right environment and the right substrate, it takes care of itself in the temperature. Shipwreck says, I spent mushroom block I was thinking about mixing into my next grow. Oh yeah, they're actually, the mushroom compost is a slightly alkaline, so it's not a bad thing, that's for sure. Popcorn time. Hey, it's Raptor. How you going, Raptor? Let's see, mate. We're talking about fungus. Yes, beneficial fungus. No questions have been coming through, but Main Green's really into it. He's experienced at it, and he just brought up some statements. So we're just sort of rambling on a bit about that. And now, yeah, Main Green, and then GMF was saying, how do we do it? Let's do it. Apparently it's easy, this stuff. And if you get the stuff from the, yeah, so my suggestion, if you haven't got much money, we'll go back up. Actually, I'll finish what time instead of saying building a substrate. Cheers, g'day. Oh, yeah. Main Green says, I grow mycelium on popcorn. Oh, popcorn is a substrate. Then I put it in cocoa and water and verum, and she newts. And some newts. Sweet, there's a good little hint. Popcorn. Did you know to grow did you know corn, there's 4 different species of corn, and one of them actually grows popcorn. So if you go that actual type, you can grow it in the summertime, produce heaps of stalks for your winter time coming up, so you can grow it in the right conditions for your indoors. That'd be pretty snazzy, wouldn't it? Raptor says, you can take a small amount of mycelium and expand it, pour some boiling water onto cardboard, let it drain, and add the mycelium when it's cooled. Yes. I'm glad you said when it's cooled, because if you poured the boiling water on when it's hot, it probably would have killed them all. Yeah, unfortunately. Probably. There's Lene. Hello, Lene. Nice to see you. We're talking about fungus today, beneficial fungus, and growing it, and talking about substrates actually at the moment, different substrates. So if I didn't have much money and I wanted to, where's the question? If I didn't have much money and I wanted to start growing mushrooms, I would probably use lawn clippings. And that would be a good source of, as long as you retain the nitrogen when it's green, you can like put in mulch piles, like just pile them up, your organic matter. And then, see, that's not going to, then you get all, you have to pasteurize it. Take it in the oven, and then you put it in at a certain temperature for a certain time, or you put it in a steamer under a few atmospheres for a certain time, and that'll sterilize it. That takes a long time. I would start it. What would I do? I'd just make some cakes and a few blends, and I suppose I'd pasteurize a smaller amount, because I got a pressure cooker, which is quite large. So I'd put a few. How's that? It fits easily a basketball in. So a basketball amount, I'd do that a couple of times, pressure cook it, and then I know it's sterilized, and then I'd inoculate that with the whatever I wanted, with the fungus that I wanted. So I'd source the fungus that I wanted somehow and get it, and then keep those spores. You can get them in syringes and plates, in on foil, on bits of paper, like from their spore prints. There's many ways you can get them. So you'd store it well in the fridge until you want to use it. So once you've gone and made these conditions right, and you've got your sterile substrate, you inoculate it, which is just add the spores to it, put the paper on top and shake it around or something like that. It doesn't take much. And try not to let the outside atmosphere get in too much, because there's fungal spores in the air, so you don't want them to get in first and beat your one that you want, to it. And it's truthfully, it's that easy. That's basic. That's a harder way to do it. You can probably even do it easier by not even pasteurizing it, because remember I spoke about that that kelp that I did a few weeks ago. I could have inoculated that when it was wet and put the spores through it, and that would have been dominant in the center and then overpowered the rest of them. So, yeah, there's the easy and the hard way. And keep it right. Then you'd have to keep it at the right temperature. So you'd have your temperature gauge and your humidity gauge around. So you want to maintain it moist, but not wet. So pretty much 60 to 80% humidity is what you want. And then the temperature, you want it to be, well, up 4 to 14 produces a fruit. So I'd be keeping mine at about 10 or 50 Fahrenheit. 10, that's at the lowest. And at the most, you can keep it, well, cool. It's going to grow rad. If it's up around like 80 or around 30 Celsius or something. I hope that helps. We'll see what's going on down here. Main Green says you can take the leftover from fruiting mushies and then put them in the garden. Oh, yeah. So the fruiting, they're the stuff that happens above the soil. So you got the stem and then the mushroom cap itself. Actually, I should be getting into some slides here instead of talking about all this. And then you can recycle, put them into your garden. Yes. So and for a spore print, it's that easy. Just cut the cap off and put the cap upside down on something, and then the spores just drop onto something, whatever you put it on. If you leave it on the bench, you'll pick it up in a day and there'll be a print there. And it's like, wow, look at that. So it's all reproduction cells. Yes, nice. Yep, you've got to pressure cook it, bro. Yes, Main Green reckons to pressure cook it. And that guarantees that you're going to get the species that you want to be thriving, and it's not going to be overtaken. Because imagine doing it the easy way without a pressure cooker, and all of a sudden you're out combated. Like all of a sudden the ones that you've had a batch and there was heavily spore dominated material you were using at the start, and then you put it into the wet conditions and the right temperature, and bang, it's everywhere, it's germinated, but you're thinking it's your one. So not to be mistaken. And I have done it in pressure cookers in the past too, when I've used specific species that I've wanted to grow, because then I've been. And then you grab those cakes. They're big. They'll fill up your substrate real quickly, and then you can grab like a handful and then put that into something. So once you've got a bigger, dominant action in your substrate, that's easier to inoculate, and then you're higher chance that you're going to get better results for your larger outdoor mushroom farm. And also the spore prints. You know, GMF from using the seeds, the little spore. Actually, let's get into some slides. I'll see what I can find here. I haven't got much on fungi, but let's have a look. I'll just share my screen and see what we get into for fungi. Fungus. There's two different types of fungus. There's your, hang on. There's yeast, which has the hyphae, which doesn't have hyphae. Actually, and there's mold, which does have the hyphae. Okay, whenever you're ready. Oh, here we go. Fungus means root in Latin. Symbiotic fungus, a few different types. I'm not really going to go into it much, but yeah, they're extremely beneficial. That's how they interact with the plants. Outside the root network, the species of hyphae and spores. So here's your root there, then there's the little hyphae and spores in the middle. So by them interacting with the roots, that's how it can form an extension of the roots. Ecto inside. Fungi, what's this? Oh, right, no chlorophyll. What? Fungi has the following features. Primary terrestrial means it's under soil, bearing spores, eukaryotic cells, no chlorophyll, obtaining nutrients in a heterotrophic absorbed way from various different ways. That means fungi does not obtain energy from the sun, and it does not use CO2 as a source of carbon. It respires it. So having fungi if you want to use fungi as a source of CO2 for your little area, you have to use 4 times the amount. So if you've got a 4x4, you want to use a 16x16, and then this will respire enough to give you what you want. Humans and fungi. No, I'm not getting that much into it. Fungi are saprophytic, means they decay things. Saprophyte, which means they obtain nutrients from dead and decaying organic matter. They've got enzymes that break everything down. That's why they're very, very important. Depends which enzymes are in certain ones too. There's ones that do certain things. There are 7 main phyla and 1 subphyla that are briefly described in fungi. Yeast, molds, fruit-rotting fungi, truffles, fungi that can cause severe disease in plants, and fungi that can coexist with algae and form lichens. Then I'm not going to get into this. Raptor will be going, no, stop. Protists, no. Look at them under the microscope. That's so cool how they look differently. I've been saying for ages about pulling the microscope out. You can watch all the little chlorophyll dance around. It's funny, and then you put like more lighting, and it goes anyway faster and slower. Diatoms, diatomaceous earth. Which is a very good input as well, organic input. Looks like it's right off a few classifications, taxonomy or classification of microbes. All right. Okay. Yes, we're all fair income off the topic here. Summary. In this module, you'll have learned. We haven't learned much because not much was spoken. The largest, it was mostly bacteria. Oh, look, there we go. If you want to test what your species is inside of your stems, you can do this test. So you'll snap you'll break the stem. I'll dry the material out first, because remember, it's going to stay inside of the material. And then you want to germinate those spores. So put the dry stems in an environment where it's 60 to 80% humidity, because remember, it's Saccharomyces is the lowest, and that's one of the lowest, and that's 65% it germinates at. So by maintaining 80% to 90% humidity, you know it's pretty much going to germinate. So that's the spores, and then it's going to tell you the color. Green's Aspergillus, so I know that that was from the Aspergillus genus, and if I was trying to get Aspergillus in there, I would have known, cool, that would have been an uptake. And you can see there, all the spores are on the outside, so it's actually gone and germinated. Then it's formed its spores again, because it's been put out of its favorable conditions, probably because of moisture. See the hyphae down here with a spore in the end? It's cool, eh? That's to test what's inside, so you can see if your beneficials are actually doing what you want them to do, and of which types. There's some spores. Isn't that cool? That's on the inside of a, that was a little pot that I made out of something, membrane or something. What else have we got here? Yeah, look at them. Pretty cool, eh? Little trichomes around them. It's cool, just a ward off protection. Oh, check out that cake. Hey, is that some gold caps? That's cubensis. So there you go. That's what it looked like. So see how that's formed like a cake. So that's cut down 4 inches and 4 inches and 4 inches. So you can see it's got the beautiful fruiting body. So it would have been in that right temperature, 4 Celsius to 14 Celsius. And then it's got all the white mycelium, and it's just thriving. It's as healthy as you want to see it, as much white as possible, because all the brown means, you know, it's not active, it's dead material. So that's under the soil. The soil level would have been there. Right there. Beautiful. That's actually, look at that structure. And that's maintained a decent humidity above it. So there's been no enzymes that have been released, and it's starting to decay on itself too. It's really nice. Let's see what else we've got here. Oh, all these different plates of fungus. So what do we got? What a shame the names aren't there, or I can't read them. But you can tell the differences. See, this one is the yeast, because there's no hyphae like this one. See how this has got all hyphae out of it? So instantly you can get down to that. This has already gone to spore. This one's formed little colonies, and it didn't like the substrate it was in, because it's actually, that might be bacteria. Because see how it's formed its biofilm around, and it hasn't proliferated, so it mightn't have liked what it was with. So that would be if you did that, I know we're talking about fungus. I always go on about bacteria. It's so interesting. It so is. Look at this looking thing. It's got like a bioluminescent. That's rad. I think there is bioluminescent fungi. Actually, there is. So that's a plate of the bioluminescent fungi. That's bloody awesome. Gosh, I could read what that says. So you could source some of that for Halloween time and just help the earth as well as our eyes. That might be bioluminescent too, but let's just say, I never would have thought to do this until reading the sign. Do not feed hallucinogens to alligators. Oh yeah, okay. That's from the Dam Week Fish and Wildlife Services. So yes, do not do that. It affects everybody. It's poison. It's actually poison. Oh, this must be a cool paper. Oh, that's where those slides came from. A journey of psychedelic mushrooms, the history, relevance to biology, cultivation, medicinal uses, biotechnology, and beyond. Those big chunkers. Richard Main Green's got some. That's not big. I can show you big. Very nice photos, isn't it? See, it's all health. And look at this in the middle on the D slide. There's popcorn. As he was saying, that's his preferred substrate. Look at that. There you go. They even do it in the lab. Oh, rad. That's a cutoff. And you can see the this would have been glass or something like that. It was cut off because it's not glass, but you can see it growing underneath the ground. So that's onto mycelium, and because the temperature is right, it fruited. So yeah, how do you know that this is dominant? So this is so tiny, you could get ones, and look at this, these spores are dead on the end, so it might be something that's come in here that's tried to compete with it, or more than likely it's probably been the oxygen and the sunlight. But yeah, maintaining what you want is the key, because there's so many spores everywhere, and if you're putting it into the right conditions, they're going to germinate first because they're in the substrate that was originally there, so they're probably used to that substrate. So the macro and micronutrients are already there for it, and then you're going to try and put yours in, it mightn't be as used to it, so they're just going to grow a bit better. This is a nice fungal dominant substrate, you can tell. It's at that temperature where it would have been getting a little bit too cold at night. It just has to touch onto that. Mapping the metabolite. Right. Just saying the different metabolites, the metabolism of medicinal fungi. Presented samples for over 50 had the medium total alkaloids of 7 milligrams per gram. Wow. I don't know how good or bad that is. I just know that take less than more, especially for unexperienced people. The person you want really, this is something that you want to have experienced people around, because you can go a bad way if you take too much. Optimum cultural mediums for fungal growth. Rad. This is, here you go, GMF if you're still watching. The best medium for fungal growth. So if you've got your relative humidity on the left-hand side and the temperatures across the bottom. So if you want to have spores, to reproduce them, so you'll be wanting to have a higher temperature. See, it's up here around 20, where see this one down the bottom is in the lower temperatures for the growth, mycelium growth, that's a little bit less temperatures. And they still love the humidity. Notice these charts don't even fall below 70%, because that's the range in which they, like Saccharomyces was 65, but that's rare. You know, not many others, most of them are above 70, and they fall into the 80, where there'll be quite a few of them. So you can see at 80% humidity here, only needs to be 5 degrees Celsius, which is 40F, and you've got mycelium growth. So that's what you've been trying to get. So you, with your substrate, you just have to come back to this chart and know what temperatures your gauges are reading. Your lowest temperatures they're reading, and your highest, and you can work out how much they're going to get. Because on the right-hand side here, it says how many millimeters per day. So 1, 2, 3, 4, 5. So you can come back and see your growth as per this chart. It's pretty rad. Aspergillus is one of the dangerous pathogens. It is common mold that can affect the growth. Also, it can actually help. There's some hormones that some of them species produce, and its spores are dangerous for humans. So see the spores here? That's the hyphae that's on it, and those spores, so I've got multiple. If that's true, it looks like it's got multiple, because the hyphae are there to carry the spores, and once it breaks off, then the spores are loose. So they can fly in the air because they're light as with wind, and it gets into our chest, and we get a chest infection. I can't remember the name of the term for the humans with aspergillus infection. Aspergillosis, I don't know, I'd be guessing. Aspergillus can be deadly. Here you go. Oh, aspergillosis. Okay, cool. So that's how it happens if it flows, if you get those spores that are loose and they come into your bloodstream. So if you're to see Anyway, I've already said the bad bits about it. Detritus growth. Hang on. I'm going through slides. I should see if there's some questions here. Turkey tail mushrooms in the endocannabinoid system. Did you know that a polysaccharide produced by Tremetes viscola, the turkey tail mushroom, interacts with cannabinoid receptors? Yes. So we have the cannabinoid receptor in our body. And if this polysaccharide peptide, the PSP, can reduce inflammation in our bodies by activating the CB2 receptor. Yes. And they're so active that our spores can germinate just about anywhere. You're talking about substrates. Look at this substrate. That's a asphalt road or bitumen road. And look, there's a bit broken out from something that cracked, and look what's coming up through it. So what happens is some spores were got there, would have flown in through the water possibly, gone into this here. The temperature and the humidity were right, and bang, it started to grow. It germinated. It started to produce its white mycelium, and then all of a sudden, bang, temperature got right and it fruited. So now they are under this cap is all those little like wing things, and that's the holding that holds the spores in. And then once it dries out, they're released and it flies in the wind and goes elsewhere and reproduces, germinates again. Entomopathogenic fungi is another one. It is rad, and I use Beauveria bassiana and Metahizium anisople, actually. Holy mackerel, it says it down the bottom. I haven't seen this slide for years. But entomopathogenic fungi, they put a disease, they put mescadine disease into their targeted insects, and there's heaps that they infect. The colors are green is Metahizium. So if you see how this is white here, this is from Beauveria bassiana, and the genus Beauveria has quite a few different species. So there's more than bassiana that you can use that I've found. And the white, mescadine disease, that's on a beetle, and that's from Beauveria. And if this was green in color, I'd be knowing that Metahizium would have been multiplying into it, because that's the effects of it. That was pretty rad. Oh, look at this. Forest light mushrooms are among 100 fungi species that are bioluminescent. Isn't that bloody excellent? So for Halloween, spin everybody out. I'd love to get some. Actually, just even thinking about the bio. Come on, Raptor. I've got to see what's in chat. He's probably saying, yeah, there's all these types. I grow blah, blah. I go bloomin'. I should, I'll leave it on. I'll just see if I scroll up. I've got my tablet open. Little stuff, clusters, what you want. That's flower newts. Looks like it's, I'm just reading chat, it's covered itself in moss. Yes, don't take 5 grams. Just come in low. Yes. Johnny, who cares? How you going? It's a fungus day today, and we're talking bio-fertilizers you just missed, which is something that you must be into, or you got to get into. Main green, 80s is good. Raptor says, Aspergillus flavus is pretty nasty, he says. Main green says, told ya. Yep, they're bad. Some of them. And this is how it happens. So this is Aspergillus, and this species is Fumigatus. Raptors has flavors. So the spores, once it's dry enough, they'll release. And they just got to get in here into our lungs, and that's the right temperature from our body, and then the humidity is high, so it germinates. And then you get aspergillosis. And this is some, how's this, you fungi heads? Yes, future fungi houses. Isn't that rad? I just love the concept. It's so cool. You could probably even build them. The walls of fungi is chitin, made of chitin. So you could probably somehow latch that and twine it together like they've done with silkworms and form a structure. That's why if you want your good insecticides and your biopesticides, you want ones that release chitinase. And that chitinase will break down the walls of the parasitic fungi. Yeah, isn't that cool? Look, that's a bit like an Eiffel Tower made out of one. That's going a bit extreme. I like this low-level stuff. I think that's rad. Skylights and everything. You can see that, look at that little observatory, little alien houses. That's sweet as. Next, fusarium wilts. There's some bad problems from fungi. So you have beneficial ones. You can also have pathogenic fungi. Fusarium wilt. So yeah, if you see some wiltings going on, everything else is pretty good. You know the substrate of that's pretty bad. Pythium's bad too. Mycetes. There we go. I support both of them. Oh, here's that spread before. So check out this poster. It's isolated that this person's made. Studying fungi is especially as filamentous nature. There you go. And you can buy a high-res version of that at that address. And that's where it'd say the names next to it. Mine aren't high-res enough to say the names, unfortunately, to get those two of the 100 that are bioluminescent. Now it's rejogged my memory. 3 things you need to know before starting microdosing. Okay, what do they reckon? Microdosing is not a quick fix. It takes time to build a relationship with medicine. Microdosing works best when used with other tools, meditation, movement, mindfulness. Yeah, because you've got to be in the right frame of mind, otherwise it enhances badness. Your intention, and the last one it says, your intention is more important than your expectations. Your intentions are more important than your expectations. So you just go with the flow. If you take the right dose, you'll get a blooming reset. Oh, no, sorry, this is microdosing. Don't expect a reset. Expect a long, gentle top-up. I haven't done microdosing. Don't know much about it. Have no experience. I just do a reset. I don't muck around. You just have a good amount. Like we were talking about before, you don't have too much. You start low. We spoke about dose amounts for Main Green and myself. So you start on those low and allow. So how I would do it is you put your keys up top. You prepare the whole afternoon. You might, you're going to have between 12 lunchtime and that whole afternoon off, including dinner time. You'll have your dinner prepared in the microwave. I'd take them about 2 in the afternoon, get everything ready, doors and windows locked, and put your keys up. And that's what I used to do anyway, and get my stereo cranked on the visual effects like Banco de Gore and some other trippy music, and have the TV on the visuals and just love it. And it'd take about, actually can't remember how long it takes to kick in, about an hour, 45 minutes. No, I can't actually remember. I haven't done it for a while. And then enjoy it for about 4 to 5 hours, and then you feel it tapering off. Then you'd have your dinner and relax and go to bed. Start up, and when you wake up the next day, if you've had the right amount, which was the amounts we were speaking about earlier, I wake up and can be thinking about a bad thing, and all of a sudden that bad thing isn't even into my thought pattern anymore. I'm just thinking about cool things, or all right, let's get onto it, or all right, blah, blah, positive stuff's flowing in. So it's like a reset. It's a brain detox. So if I remember going through bad, stressful times or anything like that, that's what I'd be turning to. Not to these bloody, what do they call them? Those, pharmaceutical types of things. That's how it works for me, though. So everybody's different. Old soil for fungi. Here we go. We're going to see what's the difference with fungi-dominated soil over time and with new soil. So why have we got a large forest here? and it's new growth. What happens underneath that? Well, the large things here have been long enough, so it's sort of like a timeline. So you can see this is growing, little shrubs. It gets bigger and bigger and starts to age. So the seeds that are in here would have been trees, and they're starting to age. So underneath it's starting to develop, and you're getting a good food soil web in here. This might be newly torn up ground, mining site, construction site, all that sort of stuff. So it's only small. So as time goes on, the fungi has a chance to take over and proliferate, and the mycelium creates as one. And yeah, you pull up all the, and go into the forest and look underneath in soil in Pacific Northwest, and it's all white. It's just excellent. And you won't find it here. Bacteria-dominated soil. So while it still has all the chances to break everything now, the carbon to nitrogen ratio would be affected where the nitrogen's a bit higher. And then once it's settled itself down, the fungi has a chance to proliferate, and that's how that happens. Oh, look at this. Guess this. I'm not going to say who it is. Raptor will get it, because I've heard him talk about this one, I think, before. All right, I was going to give you 30 seconds from now. But this is an indoor, see how it's all done very sterile-like? Because you're creating perfect chance for any fungal spore to germinate. So you want yours to go. And also you don't know if they're pathogenic ones or if they've got viruses in them. So it's very, very, this is very, doing it indoors. Oh, actually, I'm not going to say that. But yeah, let's have a look. All right, he's written a monotub. So I know that the word's out. What type we got? It starts with the letter P. Come on, that'll the letter P. That's the species. And you can see all the white mycelium underneath it. So it's just growing its cake. So that would have just, you'd put, for instance, say even popcorn. And then the mycelium would grow on top of the popcorn because it's the humidity's higher in here. So it would grow on top of the surface here, not just underneath. And the sunlight would be low. So it's not enough to kill it, the UV and things like that, the mycelium. So, yes, I see in chat, it was guessed correctly. Penis envy, P-E-N-U-S. Envy. I don't know if it's not my thing, but that's what they are. Oh, yeah, this is zoomed in 45,000 times. And you can see it's leaf covered with powdery mildew spores. So that was the hyphae on top of it. It'll make possibly the mycelium. That's the mycelium that was growing there, and it might have shriveled up and died off. It might have, this might have been cut off somewhere where it couldn't feed each other along its cells to keep the rest of it growing, and so it might have just died off. But there's your PM. Powdery mildew, another problem on top of the plant. The sources of potential microbial yeast and mold contaminants that can reduce potential harvests. So they're just everywhere. You can see they're on the air, they're on the stems, they're underneath in the substrates, they're in the water. So it's, you can't get rid of them. Even growing in the lab, it's extremely difficult to get rid of them, and yeah, you can, but once something else gets in there, it can proliferate and take over. So going organic helps, but the bad ones, because going organic, you've got a match of an even of the good and the bad, but because there's so many good microbes, the plant can be fed and maintains its cycle and growth and development functions that it wants to do, because it's all its right microbes are there. So once you try and eliminate that and go synthetically feeding, and you spray all this, you've just killed everything. You spray sub to a water drench, bang, everything's gone on the substrate. Plant might be good for a period because that problem's gone, but as for its future functioning, yeah, good luck. Just be battling, spraying and adding this. How do I get rid of that? What do I get? So bio-fertilizers, by putting in biopesticides, by these entomopathogenic fungi that are beneficial to help you. It's bit of a summary. Oh, here's a psilocybin usage study done by the AMA. So I won't go into it, but just a study, because I already know the outcomes are bloody good. That's why it's Australia is so hardcore with it. They haven't even legalized cannabis nationwide, but they've legalized mushrooms and MDMA nationwide because there's some good money in it for them. Oh, damn, where was I up to? All right, we'll go back to here. Oh, look at those studies too. Jeez. I didn't think I had much on it, on fungi. What's this one? This is some Rhizophagus irregularis fungi at different concentrations. So if you add a low dose, you can only expect so much. Medium dose, this is a root tip, the root hair. You can get some good interaction. And at high dose, you might actually get too much. There might be a problem. Depends what sort of high dose there is. But because you want everything to be symbiotic and to work into work, you might be able to get some dominant species that will be beneficial to be the dominant one. But I haven't found that yet. And one like Bacillus thuringiensis, no, that's a bacteria. I'm not going to speak about it. So what's this about? Alternations in genetic program in soil being roots from inoculation of the beneficial fungus. So you can read all that if you want. I'll zoom in, you can read it later. Press pause. There you go. We already touched on that. Aspergillosis can be deadly. Yes, the spores of fungus. Oh, look at this. This is a fungal disease. It's septoria. Blimmin' thing. It's easy to get rid of though. Like Pseudomonas florens is rad. Is just blimmin' rad. And that might have even been how I got rid of it. I don't know, but not good. That's under the microscope. So it's that. It's not. It's just. At least it's not. See, it's not infested though. So it's not. The plant puts out hypersensitive reaction. It's called within its tissue, where it kills the cells. So see how it's gotten into these little cells here, and it's gone black. So it's killed the cells. So it does. Then it'll put a ring out, and it'll start. So it'll isolate problems in the plant. So it hasn't had ability to do that yet. Oh, okay. Schematic diagram of the mechanism of trichoderma. Trichoderma is fantastic. Yeah, there's one species of trichoderma as I prefer over Hazarium, and what's the other one? It puts out hormones. Not all of them produce hormones, actually. Like here, Trichoderma variety is only solubilizing phosphorus. Trichoderma hazarium does do the fungicide as well, but Trichoderma aspergillium, look, oxen, gibberellic acid, and abscisic acid. But gibberellic acid is a very, very good asset to be used at different times. So yeah, not all of them do that. Here's a plated hazarium, Trichoderma hazarium. They put strain numbers on them because different strains react differently. So by getting, if you order the T22, for instance, because I'm into this now, you'll expect different things. So from different studies that I have, will show different things, and that's the one that I'll be trying to source. Yeah, look at this hazarium. The biocontrol activity has been reported here against several pathogenic fungi, Fusarium, Pythium, Sclerotorium. Okay, so it was tested against that to be effective. Just that one strain, beneficial fungi. Oh, look at this. The trikes on the fungi. Remember we saw it on the spores before? Look at these trichomes on it. It's pretty spiffy. I'd love to see what's in there. Is like do an extraction, see what's in those trichomes heads. It's nice glandular ones too. There's no sessile or nothing. It's all fully out, fully protecting it. Good to see what's in that. Actually, oh, far out. Main green. I've been missing out on a few. Main green says, I like the South American, the equatorial mushrooms in the mountains at certain feet where the clouds and suns look up. Sabrina, she made mushrooms mainstream, says Mayan Green. It gets crazy when you dive into plants and mushrooms, he says. Raptor says, Rhizophagus celarius is a species of mycorrhizae that is symbiotic. It sure is. Myco and Rhizo. Oh, that's the last one. Actually, well, how about, do you want any studies? Where's fungi again? I didn't realize there's so much in there. Trichoderma's role as a biocontrol, total yeast and mold levels in our plants. Magic mushroom. Okay, introduction to chemistry and pharmacology. No. Medicinal mushrooms. Is that all? No, there's not many. Let's see what this one says. Cultivation and other. There's the, oh, I thought it was a blooming website for you to get later on. Because this would probably be helping for, there's a question asked earlier about how to cultivate it. What do you need? What types? These get some other substrates. Oh, they even say corn cobs. Oh, there's some amounts. Oh gosh, this is a good conclusion to the show, I think. Don't even know how long it's been going, but there's some rad recipes. My goodness. I remember, I can't say that. Shame. Bag filling and autoclaving is a must. Yes. Autoclave, they're about $100. Get one if you're going to be interested in doing this. You'll be very, very satisfied. Once you put the material in and pressurize it, it comes out and it's all ready. Whack it in the bags, inoculate at the right temperature because the humidity stays in the bag, and look what you get on the outside. Cut the bags open when it's all fruited. That's an example. There's some different temperatures you might want to have them at and different humidity ratings. See how they don't below 65. 95, that'd be dripping at the ceiling. Okay, to induction. Well, you want to make sure it's done. 95, that's nearly floating in water. And notice all the light levels, extremely low. Temperature, days, yep. Next question. King oysters, it looks like this gets into different species now if you want to do it. And some questions for you. Observation of mushroom, yeah, it does. So that's been up the top as the summary, and then this just goes on to go through different species that you might want to get. So I'll go down here. Here's some questions for you too. Quiz yourself up. You can pause later on. I'm not going to do this stuff because it's going to take way too long. Lab and culture perfects. Yes, if you want to, use lab and culture stuff on a Petri dish if you want to, like, get new species or test it for resistances against other things. You might grab a Petri dish and put it under those conditions and put some, there's a substrate. You don't just get your Murasagi and Scooge medium with vitamins added, MS medium. And then you put your ingredients. You might want to put a bug in there or different things and just see how it reacts, different chemicals, how it reacts to it. And then you can test it on your Petri dish. So you're not going to wreck anything of your plant. There's an exercise for you to do. Look at that. There's the answers. I hope you be going to them last. In China. Very good. Stop sharing, I reckon. And now, wow. Main Green says, look up Maria Sabrina. She made mushroom live stream. And yes, that's been used a lot. I've heard of that one. Those bags are reusable. Oh, good on you thinking about the earth. Yes, reuse those bags that you cultivate in. I save all my bags, like, you know, out of the cereal packets. They'd be perfect for those types of things. You'd go and you'd have to clean them well and truly, or even they've got a lot of sugar on the inside, but you don't know what spores are on the inside, so you'd have to put them through the sterilizing process. But yeah, well, write your stuff in chat now if you want, because thank you for those who have really helped with the fungi, and I can change today's title to Fungi, Bio-fertilizer, what all the different things, dose amounts, culturing, growing, temperatures, moisture, different species, safety. Actually, I put that up at the start because I always like that. Yeah. And did you know too, it's a bit of a shame that they won't, I've been with YouTube for a while. There's a special rating, EDSA approval for education and scientific things. And I've been trying to get that. And if you want to help me a lot, you can write to YouTube and just let them know how helpful and scientific this is, and how I tell about the bad things and the harmful stuff, as well as the positives. And it's not about street recreational blooming stuff that they reckon. I even try not to swear. You know, it's I really do try hard. So if you really do want to help me, that will help me, because I'm just sick of battling with YouTube. I've just gone back and forth so many times for that. It can be, here Raptor said, just to conclude too, those bags, you can use them up to 3 times. So that's some good information. So he's speaking from experience, that fella. So that's really good to know. So don't throw them out like they say. Pasteurize them and sterilize them and put them through the processes again and reuse them, and help Mother Nature. And she will help you by giving you more fruit. Yeah, so I'm glad I said that at the end, because I might say that at the end or try and bring that up a bit more often, because there's a few tech people out there and they can just write to some people and do the correct things. And, hey, why is this getting struck down? It's so educational, or I don't know, something along those lines. That's how I tried to portray it anyway. Yeah, and from my studies, I think it comes out really well, because seeing what other people, I'm not going to compare to other people, but I just try my best. So today's show, put it in chat now if you want anything else said. Please, there's a 30-second delay. So today's show will be getting retitled Fungi Special. Very good. Thank you for those who listened, and thank you for your ears. Thanks for those who've written in the chat too and made it a special so we could discuss all these cool topics. And I hope it helped those that needed some help. That's what I always enjoy too. Helping others is the main thing. It's not bro science around here. It's real science, university science. Stuff that I learn, and I start my uni next week. Woohoo! I'm so excited for it. 2 microbes, advanced microbiology and microbiology technologies, how we can control microbe environments using microbes. And the 3rd course is, I've done it at plant metabolism and physiology. I've done it from another university before, but this one was offered from one that I haven't done. So there might be some new up-to-date information. The course was, I think it's only 1 or 2 years old. So I look forward to doing that. Might be able to learn something and, yeah, gain something out of it. It's long as I get one thing out of a course, I consider that an excellent course. And one thing can be so little, but it can be so beneficial. Renee says, love the mushies. AussyCC, especially the bioluminescent. Yeah, 100 different types. Fair dinkum. Why hasn't people got more of them and put more fixtures of them around? I'd actually like to nearly get some spores and place them around the yard and stuff. How rad. You don't have lights at night. You got mushrooms. How cool would that be? Yeah. Not cool, YouTube. Yeah. Yeah, especially when I try. Because I don't get many views and things like that for the amount of effort that I put in. And I feel a lot of people would benefit from this information because it's up-to-date, relevant information. Because my university studies, there's only, well, I still study, and some of the courses were written this decade. So it's cool. Yeah, source some of the bioluminescent mushies. Let's get some more information about that, because we can have the spores and they are legal. They just look cool. So let's get some. Righto. I appreciate you. I want to say everyone for listening again and writing in chat, because without you, it's not a show. And still 8 people. So fantastic. Thank you. Whoever use 8R. We can see next week. Next week's on again, and let's see what is going to be for next week. There's no topic, so you can bring me questions, whatever it may be, related to, my topics, plant science, soil science, microbiology. Love it. I'll have some slides, hopefully, like today, even though I showed them a bit late, didn't I? But we still got there. Raptor says, you can use mushrooms for food and medicine, give the waste to compost worms, then take the worm castings for your plants. You're welcome, Mother Nature. That's exactly right. Sustainability, helping the environment, and feeding everything along the way, keeping that food cycle web cycling. Very well. Nice words wrapped up. Good stuff. You just got that in time, mate, because it's been 1:12 now, and I think I'll let you go, and you can enjoy your day or your evening and save all your questions for next week, and I hope to see you all again then. Thank you again for listening. Happy breeding, happy growing, and good health to you all. Bye-bye. That's a good idea. Then I'll end now. So I should wait 30 seconds afterwards, and then I'll end. Okay. Use 3 folk just gave me a new technique at the end. Now bye. See ya.